So I'm here. Hi, hello everyone. Hello and welcome. Microbehunter here. This was a little bit a confusing start for me. I have to tell you because I did not quite find in how to start the live stream now. That's why there was a little bit of a delay, but I think it seems to be working now. Okay. Yes, I can see myself here. Okay. Yes, that's good. Yeah. Okay, what a day, what a day. Let me see. Yeah, 38 people are watching right now. I'm just a little bit of housekeeping like every time. Give me a second or two or three. Okay, I think we're ready to go. Loud and clear. Okay, hi, hello. I almost cannot believe it that I made it. Yeah, I had a little bit of a technical issues today with my computer. The network didn't quite work. Connection, router problems, hardware issues, but everything seems to be working right now. I'm happy that you're able to join me again. Yeah, I had a little bit some thoughts on what I could do today, and in the morning today, morning my time, I thought, well, maybe I'm going to do a little bit of soil microscopy, but I changed my mind. The reason is because I actually did prepare a specimen. Here it is. Yeah, but honestly, some soil water, but there was not anything. There wasn't anything interesting to show you. So I'm still working on this a little bit, and I decided to change the topic around, and I would like to try something today. So I would like to try this stain here. It's called SAFRANIN, and I would like to stain a few things, and we're going to see how well this actually works. I have just made the solution today, so I've not been using it quite a lot yet. So many of the things that I'm going to be staining today, I'm just actually going to try out the first time right now here live. Okay, so what I'm going to do now is I'm going to go through all of the comments here to say hello all around the world, and for a couple of minutes we're going to look at last week's slides, how they actually how they look like. Okay, because if you've watched been here last week, I made slides using different mounting media, and now I would like to see how if they're still usable or not. But I'll quickly go through the comments section. Okay, just a hello. It's a little bit of a tradition to say hello all around the world. Hello Athens, London, Netherlands, Hungary, Netherlands, Texas, United States, Belgium, Oregon in the US, Germany, northern France, Philippines, Tennessee in the US, okay, Montreal, Georgia, okay, and the UK in Essex, sound and video good, okay, very nice, from Germany, okay, and from the UK, from the UAE, I think, New Zealand. So Finland, isn't this cool? And yes, okay, that looks quite nice. From India, okay. So, and from Turkey, very nice. Okay. So, yes. So, I'm going to change if you want to ask any questions, please feel free to comment in the questions in ask questions in the comments section. Here we go. After approximately 1 hour, I'm going to go through the questions again, and what I've done also is I'm collecting questions, and every Sunday I'm going to upload approximately a half an hour long kind of podcast where I'm just also answering questions. So you'll find this also in the channel here. And some of the questions I might be reusing again, and I will be answering them separately again. Okay. So, how are the slime molds doing? I will also show them to you. Why not do this now? Then we're going to have a look at the slides here, and then I'm going to do a little bit of staining. It's a very spontaneous live stream. So if you have any specific suggestions or topics, then basically I will always pick up on that as far as I can do that. So there are a couple of slime molds here, and if you don't know what I'm talking about here, you might have to go back a couple of weeks. And I have been growing slime molds on filter paper, and I deliberately let them dry now because the dry form is called a sclerotium. These are cereals here, and basically they should be able to store for a long time. And if you put them again into water, then they should come back to life. Yeah. So actually, they've been growing quite well. You don't see it a lot here. There is a little bit here as well. So I've deliberately allowed them to dry. Yeah. Also, this is not so nice here. I tried also different types of food here, and you can see that yeah, it also started to dry up, and this is basically is now also something that I can store for a longer time. And then if you add water, then they're coming back to life. This one is a slightly more fresh sample, or is still fresh. I put cereals here, and they were kind of moving along, yeah, from food to food, and now basically they found this cereal grain over there. Okay, yeah. If you want, I can quickly put them under the microscope, stereo microscope as well. Okay. Just to here we go. Good, very nice. Yeah. So yeah, so that's basically the slime mold. Yeah, and you see it's still going and healthy. Yeah. So yeah, it's quite a nice thing. I'm quite happy that this actually works out that well, and I think those slime molds are really nice specimens, specifically if you have a stereo microscope. So these are the old cereals here. You can see that they already look very digested. Yeah. Yeah, and they move away once they started to digest it, and then they move away and they're looking for something new, right? And as they eat, the slime mold starts to branch out more and more and starts to become larger and larger, of course, because yeah, it eats up the food and converts the food as well. Yeah. Yeah, there's a suggestion if I should not put some of them into the freezer and see if they are able to survive. Okay. Yeah. So, and can you tell us how you made this time lapse of the slime molds? You know what I will do is I will, as a matter of fact, I will answer all of those questions later on. I do want to save a couple of questions for later on as well, and also, of course, now for the new podcast, so to say. You see, I'm experimenting around quite a bit with different formats as well. So then, before we do the staining, let's put this here away. Let's have a quick look at least at those slides here. So for those of you who don't remember or who did not join in last week, I took some pond water samples, and I made slides using PVA glue. That's a water-based glue, glycerin gelatin, and also Euperol, which is a non-water-based one. And if you put this, no, I'm just going to show you those two here first. Because they're quite different from the other one. Let me turn up the light and go to the scope view. I have to move up here. Okay, and of course I have to find the focus. And what I'm going to do now is I'm going to remove the prism to give it some natural light. Not based on natural, yeah, basically the natural colors rather. Okay, and these what you see here, that's mounted now in a water-based mounting medium. That's PVA glue. You see a couple of air bubbles, but you see that inside those cells, these are diatoms. Okay, there are the green chloroplasts, and where's the arrow here, right? So these are basically the frustrules. These are the shells of the diatoms, and inside you can see that it's green. These are the chloroplasts because they do photosynthesis. These things here are, of course, air bubbles, right? And this is basically mounted using a water-based PVA glue. And yeah, it looks more or less naturally, basically. Yeah, of course, there is no movement. Water samples generally are quite nice when you see the things move around. But this one generally is okay. Yeah. Glycerin gelatin, a similar case. So I'm going to put that in as well. Now, luckily, the camera again works. I'll readjust this. Okay. And yeah, that's basically in a glycerin gelatin. And you can see that it looks very similar. Yeah. So water-based mounting media means that the colors, the pigment, the chlorophyll still stays inside the cells, right? Because the chlorophyll does not dissolve in water. So, but now let's take the Euperol mounting medium here, which is based on a solvent. Let me carefully take this here out, put the other one in. And let's go up again with the magnification. And the situation is here quite a bit different. By the way, I just recently, a couple of days ago, I actually made a short video about this. And what you see is different is that most of those diatoms here, they basically have now lost all of their color in here. I mean, some of them still have retained it, but most of them seem to be empty. And the reason is because the solvent in the mounting medium around it dissolved the pigment and it kind of leached out. And you look at the background here, the background also looks a little bit yellowish, greenish, brownish Yeah, because that's essentially from the color of the of the chlorophyll that leached out. Yeah? So you see that it looks. Actually, I have to admit, it does also look kind of nice because it looks a little bit more cleaned up because now you see the individual shells of the diatoms much better, right? Yeah, because the solvent has removed all of the other structures. Yeah? So I just wanted to show this to you. Yeah, basically what has happened over the last week. Yeah. So as the mounting medium starts to dry, it of course there are changes happening here as well. So what I'm going to do is I'm going to so slime molds. Okay, slime molds. Okay, I've made a slime time-lapse of frog spawn development. Ah, very nice. Okay. Maybe UPRL is ideal for ocean diatoms. Well, let's maybe. There is a specific mounting medium with a right refractive index that's actually commonly used for diatoms. And I don't have that mounting medium, so I was just experimenting with those here. UPRL actually is the mounting medium of choice for insects because it clears the exoskeleton of the insect. So basically, insects generally are very dark, but when you use a UPRL mounting medium, then the exoskeleton becomes much brighter. So what I'm gonna do today is I'm going to do a little bit of staining, using SAFRANIN. I just want to show you how and where I got that from and how I made that. Well, I ordered from a British company from the UK. It's called Brunel Microscopes. I already mentioned them before, and they basically sell online pretty much almost everything that you want to have. And stains are also sold, and many of the stains are actually sold in powder. There's really not a lot in there. It's maybe a gram or something like that. But it's way more than you need, and it's fairly cheap as well. And they're allowed to send powder around the world, but not liquids. Also, Amazon doesn't send liquids around, and other shops neither. So the mail system has become kind of restrictive about sending liquids, but it's not a problem of actually sending the powder. And SAFRANIN powder is used for also staining bacteria. And I've used it also during my university studies quite a bit when I did gram staining. And it's also a general purpose stain. And what you have to do then, of course, is you take the powder and you have to make your own. Staining solution. And I've done that by basically making a 1% solution in water. That's why I called aqueous. Means I added 30 milliliters of distilled water and 0.3 grams of this of the stain of the powder. And you see there's still plenty left. It's more than enough than I'll probably ever need. And I mixed it, and basically it looks red like blood. It's a really powerful stain. And what I'm going to try now is I'm going to try to stain my own cheek cells. And I also would like to stain some plant cells here. Maybe you've already, I'm always come back to those again. They're in alcohol. So that means the green chlorophyll has also been removed now. And the third thing that I want to do is I want to see if I'm able to stain the cells of a tomato, right? So we're going to see how far, and a piece of wood I've also got. So we're going to see how far we're going to get, okay? You already know it's highly experimental, the whole thing. This means that in many cases, or in some cases, the things that I want to show don't work. Okay, and we're just going to take it from there. And I'm actually already looking for something, and I don't know where I put it. Just a second. That's a little bit. Here, over there, next to my microscope over there. I'm going to start by actually trying to stain a couple of cheek cells from myself. And what I do need is I also need a container for the wastewater. Just a second. Wastewater, because I'll be rinsing this again. And what I want to do now is I'm going to do the following. I'm going to collect some of my cheek cells from the inside of my mouth. I'm going to transfer it on a slide here, and I'm going to heat fix them. I've got a little alcohol burner here to bake them to the slide, and then I would like to use a SAFRANIN staining. So honestly, this is the first time I'm doing this with SAFRANIN. Okay, so yeah, we're gonna see how well this actually works, and the point why I'm doing this is because I just want to demonstrate to you also how this microscopic technique works. Okay, so the first thing that I have to do is I have to collect some of the cheek cells. Okay, you kind of, yeah, almost like brushing your teeth. So, okay. So, and yeah, I just transfer them here now on the slide. Okay, there is not a lot of liquid here anyway. And what you have to do is, yeah, you can actually see, obviously, if you just hold it against the light here, there's something there. And what you have to do now is you actually have to, I'm going to throw this away. I've got a little trash can over there on the table. And you have to now wait until it dries. I mean, this is going to be very quick anyway, right? There's almost no liquid here. It dries very quickly. And what you then have to do is you have to pull it through a fire, a flame. You don't want to burn it, obviously, with the cells now pointing upwards. Okay, so away from the flame. And by heating it over the flame, we are basically baking the cells to the glass slide. So it's almost like an egg that sticks to a frying pan. So what happens is the proteins in the cell, they form cross linkages to the glass. And so that they cannot be washed away by the staining process. Okay, so that's a little bit the idea. So you see it's already dry. Yeah, and now I have to heat fix them. By the way, folks who are actually into bacteriology are also going to heat fix bacteria. And what I have to do now is I have to move this away. Here there's alcohol in here. So I want to move the alcohol away so that the whole thing doesn't go up in fire. And the second thing that I have to do is I have, you see there's a camera right above yeah, here. I don't want the flame to hit the camera. So I'm going to move it further away. Okay, like this, so that, yeah, so that's going, yeah, I think it's going to be fine. So it's now clear from the camera. Okay, so that's the slide here, right? And I need to kind of see if I'm able to make a little fire here. Yeah, it'll work. Don't worry. I know this. Yeah. See, you just have to wait a little bit until the, yeah. So the thing is for safety reasons, I don't like to have gas in my apartment. So for safety reasons, I just bought myself an alcohol burner here. Yeah, and the flame is also not very big, but it's enough. It does its job. It's kind of small already. So what you do is you kind of, yeah, with the cells pointing away from the flame, you just pull it through the flame. How hot should it be? It's already enough. It should be just hot enough that you're just barely able to hold it. Okay, that was already way more than enough, and we're finished. Yeah, that's it. Okay, so now the cells should stick. The cells should stick now to the yeah. I'm cooling it off now. Should actually stick now to the glass. Okay, and even before staining, I'm just going to put it under the microscope so that we just have a look at it. Okay, and then that we have a before and then after comparison. So let's put everything away here. So here we go again, and no cover glass or anything. We're just going to do a very quick yeah, check, and yeah, let me see. Yeah, see that's basically regular bright field. Yeah, so I mean I've got a pretty fancy microscope with DIC and whatnot, right? But when we want to do staining, then we always want to use the regular bright field. There are not a lot of. I've not reached the place where there are a lot of cells here. Here we go. Yeah. So the epithelial cells from inside the mouth are pretty flat. Yeah. Yeah, and yeah, here they are. Here's actually there should be more. Yeah. No, these are not. They don't look quite nice here. I mean, the one that I had before was actually quite nice. This one, that's a typical one. Yeah. Yeah. In DIC, no, not DIC. Let's use phase contrast. Yeah, it looks like this. Okay, so that now you see the importance of phase contrast because it really increases contrast. Yeah? But there's only one cell. Here are two of them. Okay. Well, it looks almost like a face, like an embryo. Here's another cell. Yeah. I just yeah. I also washed the slides before to make sure. Let me quickly check. Yeah. Let me quickly go over here. Just want to check in person. And here more cells. I found more cells. They're not. Honestly, there are not a lot of them here. Well, here there are more. Okay. Let me see. Here more. Okay. So that's basically how it looks like in regular bright field. This is how it looks in DIC. Okay, and what we're going to be doing now is we're going to just stain them. Okay, so let's have a look. Let's take this away again, and the desk view, and now we can move it back here. And is it focusing? It's focusing. So I'm now going to put a drop of this staining solution on here. Yeah, we have already last week talked a little bit about those blue, that's crystal violet. Look, I also, this is not blood, this is SAFRANIN, right? It's also such a strong stain that yeah, it's difficult to remove. Have not tried the different solutions yet. So, but let's give it a try. Okay. So I'm going to add a drop on here. I'm going to close it again, and I'm just going to wait a minute or so. Okay. Maybe you know what I could do is I could actually take a tip from a pipette and spread it a little bit over here as well, so to make sure that all of the cells are lightly covered. Okay, and I'm going to wait, for a minute or so, and then I'm going to, and this is now going to show you why it's important to actually do the heat fixing, because I'm not just going to drop a cover glass on it, but rather I'm going to wash off the stain again. I'm going to try to wash it off, and the cells should still stick to the glass slide. Okay. Yeah, you know what I'm going to do? I'm just going to use the time here. There were no soot marks in all during the heat fixing. Yes, I'm using 96% alcohol. That's I often get soot marks when using ethanol. Really? Okay. No, I mean, I'm using actually a so-called denatured alcohol. So it's just the regular alcohol that you use, the cheap one, because there's also an expensive one, which is more expensive because of the alcohol tax, right? That's of course included. But the one that I'm using cannot be consumed because they added a substance that makes it not drinkable, and that's why it's significantly cheaper. But I don't, to my experience, this additive that was added did not cause any formation of any soot or anything. Yeah. So let's try it. And again, this is going to be the how do you say, the exciting part, because I don't know if it's going to work or not. So here it is. You take a wash bottle and you carefully, I really hope that this works. Okay. If not, I'm going to do that again, and I'm not going to use heat fixing. And let's see, I need, oops. Okay, I just realized I don't have any tissue paper here. Okay, I'm gonna get some from down here. Okay, so let's try it. Again, this is not blood here. Okay, that's SAFRANIN. Okay, and so I'm going to try it without a, you know what, no, you should always put a cover glass on top because it improves the resolution. What's Cymol? Cymol is a disinfectant that is sometimes added to glycerin gelatin to prevent the growth of fungi, to make it, yeah. So let's put it on here. And now I really hope that the cells are still around. Otherwise this is going to be a demo. But that's actually also one of the things why I like doing this stuff. And it's a little bit improvised. Not everything has to be perfect. Okay, not everything has to be perfect. but I think you get the idea that actually the staining did work, right? Yeah, so here we go. Yeah. So that's yep, that is actually very nice. That's actually what I wanted to show you here. See, I'm very happy that yeah, the demonstration actually worked out. Yeah, so what you see over here is the following. Obviously, you see the cheek cells, yeah, stained red by safranin, and they're still there. Why are they still there? Because I heat fixed them, right? Otherwise, they would have been washed away by the washing process. Yeah, so that is the thing. And sometimes, what you're going to be, maybe I don't know, if you're able to see some kind of red tiny dots here on the side, then very often these are also bacteria, because there are plenty of bacteria in your mouth as well. Okay, I've in the past, I've always used methylene blue for staining. It works. Look, very nice. You see the two nuclei here and two cells. Let's go up a little bit with the magnification again. Yeah. Ooh, what was this? Is there something crossing the swimming across here? Okay. Okay. Ah, no. Yeah, there was something swimming across, but that is not. Yeah, it's just capillary action. Yeah. So, yeah, so that's basically two cheek cells stained in safranin. Yeah. So that is quite nice, huh? That this actually worked out the way I wanted it to work out. Here's I don't know, another. Yeah, so these are definitely cheek cells. Here, many of them air bubble, right? And of course, yeah, you can see, yeah, the cells all have a nucleus in them. Yeah, so these are actually one of the easier specimens to microscope, and it's actually how do you say, a simple technique here that I also wanted to show you, not only staining but also heat fixing, which is a common thing. By the way, if you wanna, I'm not gonna do that now, but if you want to use oil immersion microscopy, the 100 times oil immersion objective, then what you do is after the washing, you allow the slide to air dry again. Okay, and then you put the slide under the microscope with some oil directly on the specimen, so no cover glass. Okay, no water, no cover glass. Directly on the specimen, you put immersion oil, and then you can do oil immersion microscopy. So that's another reason why you do the heat fixing, because then the oil from the immersion oil does not wash the cells away either. Yeah. So there is no need for oil immersion. There is no need for a cover glass. Yeah. Okay. Could you do a second stain on top of the safranin one, like a combination of methylene blue and safranin? Would that make sense? I have no idea if double staining in this case would make sense or not, and what the result is, and I have no idea. So what do we do if we don't know, and if you want to know? We try it out, right? Okay. So what time is it? It's already half an hour. So what I'm going to do is I'm going to recycle the same slide. By the way, thank you for all of these comments. That's actually the whole point of doing this here, simply to experiment a little bit. So, and what I'm going to do now is I'm going to carefully going to remove this one here again, and I'm going to simply add. Still a little bit. I'm going to simply add some Löffler's solution. A Löffler's solution is methylene blue, and it is already, basically you don't have to wash it away. You just can, because it does not, because Löffler's solution, I'm just looking here, I can actually, acetylcarmine, that's just a regular ink. Fountain pen ink also works. This is Löffler's. That's the one that I'm going to be using. What else do I have? I have also here crystal violet. And what else do I have here? Here. Yeah, iodine, of course. You always need iodine if you want to test for starch, right? The microphone, put it over here. Yeah, so that's iodine, right? So that's basically, and I'm going to now just use Löffler's, which is methylene blue, plus alcohol, plus potassium hydroxide. And I'm going to put it on here. And cover glass goes on top, and I have no idea how this is going to look like. Yeah, maybe too much, I don't know, maybe a little too intensive. Let's remove the excess so that also the cover glass does not start floating around. Okay. Let's have a look. I have no idea. Really, I really have no idea. Never done this before. Safranin and Löffler's. Maybe one of the two stains is stronger than the other one. I don't know. So, yeah. Let's have. Oh, okay. Yeah. Okay. Look, it's a little bit red and a little bit blue. Yeah, but the blue seems to be stronger. Also, if you look at the background. Yeah, but it's a purple. This could be some kind of, what is this? Some kind of a fiber. Ah, that's actually also quite nice. Yeah, do you see those things here? Those long ones here? What could that be? Those little, yeah, these are of course bacteria. Yeah, in the mouth there are many bacteria. So, yeah. So this is, yeah, it's interesting. Yeah, it's a little bit of a, yeah. It seems the contrast is definitely higher, no question. So, and here you again can see that there seems to be some kind of a mix between red and blue. So quite interesting. You see, there is there are no limitations here. So okay, I'm quickly. Could you not add a diluted drop of saffron into the unheated sample? Yes, I can do that. So the question is why do you have to heat fix it? Can you not just do it right away? Yes, you can. I've actually also tried that. This is something which I did try before. I'm going to show this to you. Yeah, it is possible, of course, to simply add the saffronin directly to the slide. However, you will see that the background is then much more red, which isn't a problem really, but the contrast is not quite as high. Okay, how does staining affect? Okay. Okay, let's give it a try again. The fibers, of course, that we've seen was from the cotton swabs here. Okay. So this is basically number one, and where is safranin is over here. A tiny drop. And we're going to directly put a cover glass over. Of course, when mixing your own stain, you can, of course, adjust the concentration of the stain as you like. And in my case, I think it's probably more on the more concentrated side. I always have to check if it's only one. Yep, it seems to be. I don't know, it feels so thick. But actually, it's only one. Okay. So, very dark, as you can see. Let's remove this again. Yeah, so, again, this is not heat fixed. It's a direct staining. And we're going to see how that works. And the reason why I heat fixed it is so that I could wash it here. Now I'm not washing it. So let's see how the contrast looks like here. Scope view. Ah yeah, here we go. And that is basically now the one. Yeah, with the, let's go up with the magnification. Yeah, this is gone. This is A. Yeah, and you see, it does take up the stain. So you see that the cells are darker, red, right? But I think you get the idea that because the background is so red as well, we don't get quite as much contrast. Yeah? So this is actually a good suggestion. Thank you for suggesting this, because this is actually demonstrates the importance of the washing. And the methylene blue, at least the Löffler solution, if you do a direct staining, then the background is not quite as blue, interestingly. Yeah. Yes, it is. There was also a question from Paul, if it's possible to mix stains like methylene blue for acids and iodine for starch. Yes, of course you can do that. Yeah, it's called also differential staining. Yeah. So yeah, so this is kind of were the cheek cells, right? What I would like to try now is, and again, this is again something I have not tried yet with safranin, and that is, look, oh, this looks kind of nice. That's an ear bubble. Ah, look at this. That's interesting. In the ear bubble, yeah, the cells are actually now much better visible. Yeah. Yeah. It's not quite as, the cells are not quite as clear maybe. Yeah, it's okay. Yeah, because of the differences in refractive index and some kind of a diffraction patterns are there. But yeah, so that's basically inside the ear bubble, and that's where there is the safranin still around. Yeah, so you see the importance of this washing step. Yeah, so what I'm going to do now is I'm going to, and the next one, that's going to be a little bit of a how do you call this, a dangerous thing, because I'll be working with a sharp blade. But I heard that essentially safranin is also commonly used for staining cell walls. Okay, so what I would like to try now is I would like to cut and I would like to cut and stain, and for this I need I now need a proper surface here. I would like to make cross sections of a stem of a flower and stain that with safranin. So what I've got here is the following. This is a very old sample. I've got, I don't know, I made this back, I don't know, 1998 or 99. This is a long time ago. And basically this is just a flower, a tulip. And I basically put it into alcohol, the alcohol removed the chlorophyll, so it became white, and then exchanged the alcohol again. Yeah, and this has been storing now for well over 20 years, and I'm still using it every time when I want to look at a plant sample. I take a small piece from here, and yeah, and there's still plenty enough left. And what I do is I simply go in here. And let's pick out a stem here. And the thing is because it's alcohol, when it dries, quickly, then it will shrink quite a bit. So just for your info that you're not surprised about that. And another thing that I would like to do is I'm going to now take another slide and I'm going to press it over here across. And now I need to put on my glasses because I have otherwise some vision problems. And you just press it down, and I'm just gonna slice it off here. I just realized one horrible thing, and that is that this razor blade is totally dull. See if this works. Okay, it's better. And I'm going to try to make a very, very thin cut here. I don't know if I can do that. Yes, here it is. Okay, here I've got a fairly thin cross section, kind of. And let's do it like this. On here, you know what? Let's give it another try. And so another one here, and so let's do another cut. And here it is. This one is even a little bit thinner. Okay. So I know I'm always looking down, so I know the camera's up there, right? But unfortunately, I have to look down because I'm not able to look at the monitor. So I take this stem here and put it back now into the container. Okay. And then it will start to swell up again. And before this one dries completely, I'm going to now add a little bit of the safranin. Here it is. No, I'm not going to I'm going to do yeah, you know what. I'm going to divide this. Let's do a little comparison. So one I'm going to add is the safranin, the other one maybe a little bit of methylene blue. How is that? Just for the fun of it. Let's try 2 different stains. Or 3. I don't know. You know what? 3. Why not 3? So the first one is safranin. The second one is, the middle one is Löffler. Where's Löffler? Here is Löffler. It's the methylene blue one. And you know what? Just for the fun of it, the third one, let's take another blue one. Let's take crystal violet. And yeah, you see it's significantly darker. And what I would like to do next is I'm going to remove all of the excess stain, either using some tissue paper, and then we're just going to drop 3 cover glasses over it, and we're going to take it from there, I would say. And in the meantime, while I'm looking at this, I'm going to check again some of your questions or comments. Okay, could a second stain? Are there any substitutions for safranin? Substitutions, well, let's put it this way, different stains are used to stain different things. I mean, what I have also done is, I've also made videos, is I've got another one over here that's carmino acetate, which is also extremely blue. I mean, generally, it depends. There is no general solution here. It depends really what you want to stain, right? And if you want to do a gram staining, then the standard is that you use safranin. Yeah? Is there some simple way to know what to avoid with immersion objectives? No cover glasses so we don't damage them. Let's put it this way. Oil immersion microscopy is something I generally don't like doing so much. Okay, I know this does not answer the question that you're asking here. And one of the reasons why I don't like doing oil immersion microscopy so much is because it's a little bit messy, right? So generally, what I would say is I'm not so much worried about the oil immersion objective. I'm more worried about the other non-oil objectives that might become contaminated with immersion oil. Because chances are pretty good that you're going to forget that you've got immersion oil on it, and you're going to rotate the other objectives into the oil, and then yeah, you have to clean them. So I think that's enough for the stains. How are we going to remove that? That's the next question. I could theoretically do the following. Let's try different systems. I could actually pick them up with a pipette. Okay, go like this, and we just basically remove all of this excess, stain. Okay, so that's one thing. Let's always exchange the tip as well. The second one was Löffler's, which actually we don't have to remove because it's fairly bright anyway. And the last one is the crystal violet. I had a lot of stain here. So, and what we do now is let's do a little bit of a small washing step. So I've got a little beaker here. I'm gonna add a little bit of water, and let's do a little bit of washing. So here, 1, 2, 3, a little bit more maybe here. So, and I'm going to put this. You don't see that I'm actually putting the liquid into the beaker over there. Yeah, I guess I could use the same one here. There is almost no. Here we go. And then let's see which one actually worked best. I know the samples cannot all be compared because one of them is, I think, a little bit thicker or thinner than the other. So I know it's not an entirely fair comparison here. But this one here is still quite dark. So I'm going to add a little bit more water. You see, that's actually one of the things that I did not plan doing, is actually comparing the stains. But that's fine, you see. I actually just wanted to try different things with safranin. but this crystal violet really seems to be quite strong. Okay. I think it's gonna be enough. Well, I think as a proof of concept, it should be fine. Let's just drop cover glass, one cover glass on each one of them. Where did I put my cover glasses? I lost them. No, I didn't. Where did I put them? I mean, I got the big ones over there, the round ones over there. Here they are, right, right in front of me. So, one. I know what you're thinking. You're thinking, why am I not adding any water? Don't worry, I will. Don't worry, I will. I sometimes like doing that later as well. I'm just doing it like this so that the specimen cannot go away. And over here, 1, 2, 3. Some of them seem to be a little bit very thick. Maybe I'm gonna add another one over here. And yeah, I honestly, I know I'm always looking down. You're supposed to have eye contact with the viewer. I know all of that theoretically, and I don't know how to solve that problem. And maybe I'm just gonna install a camera down here. I have no idea, so that I'm always looking at you. So, but we now can have a look at the thing here. Just a second. I'm going to switch over to the microscope over here. So here we go. Let me start. So that's the first one. That is the, where's the saffron? Where is it? Ah, yeah, that's the SAFRANIN one. Okay. Ah, yeah. Okay. That's how this is how it looks like. So I'm 10 times objective. You know what? I'm going to, so that you see it better here on top. Yeah, that's the 10 times objective. Yeah, some air bubbles in here, but I think that's fine. You see that, yeah, there still seems to be some kind of cell contents left because even the center of the cells stain red. You can see over here the edge. Okay, so that is the first one. The second one is Löffler's. This looks actually quite pretty. Okay, maybe this looks better because I was able to manage to get a thinner cross section. This could be it. Okay, look. This actually looks quite nice. Actually, I prefer this one over the other one. But this could indeed be because of the thinner cross section. Look, I think it's like this, that those cells that seem to be those cells like here over here that seem to have this blue content, yeah, apparently were not completely cut open, and those cells that are white here, they have lost all of the cells contents, and therefore they are white, and some of them still seem to be a little bit intact. Yeah? So that is the Löffler's, and now crystal violet. Ooh, this is a dark one. Let me turn up the light here. Maybe also because it's a thicker. Is it thicker maybe? Oh, it's over here. Yeah. You see, Yeah, so what we see here is definitely due to the intensity of the stain, which is slightly purple even here. Also, the background is not quite as white as the one from Löffler's, but maybe mixing it, we are trying out different concentrations. But I mean, we do see that the cell walls here are definitely stained, right? The cell walls here, right, are definitely stained blue with the crystal violet. In here, where there are no cells, it does become brighter, but you see it's still losing a little bit of color here. Yeah? Let's go back again to the SAFRANIN one. Ah, yeah, okay. Here it's also, maybe that takes a little bit of time for the structures to absorb the pigment. Let's go down a little bit with the magnification here. Okay, so that's SAFRANIN. Also looks kind of pretty. Over here again, the methylene blue, Löffler's, which I also cut significantly thinner. and the crystal violet one here, which is way too strong. Yeah, so probably I should have washed it a little bit better. Yeah. Yeah, okay, but maybe it gives you a little bit of a first idea of what you're able to do here. Well, actually, saffron is also quite nice. So let's have a look. Let's have a look. How does staining affect phase contrast illumination? Well, let's try it. It's a difficult specimen, okay? So let's use the thin one over here. So I'm going to try to switch over to phase contrast, just because you wanted to know. So, but the problem that I have with phase contrast is I've only got the high magnification 40 times objective, and that's how it looks in phase contrast. Okay, so it's an overlap of the stain, and the phase. So that's is how it looks like. The typical thing here in phase contrast is the background is a little bit not white but grayish. So that's basically a combination of phase contrast and staining. Let me quickly go through here. A little biology lesson, please. The difference between cell wall and cell membrane. That's a long one. I'll answer this later, okay? In the second part here, because it's a little bit off topic, or I'm going to make a video. Why not? Okay. Why not use the hand microtome for good stem section? You can do that. I have done that, but it takes more time. And for the sake of doing a quick live stream, it would take too much time. That's the only thing. Yeah. And for our purposes, it also worked. Yeah. So can you put some alcohol after the water as well? This will decolorize it a little bit more. What I will do, yes, I want to try this here maybe. Okay. And then I would like to actually stain a little bit of wood. Okay. So the decolorization, let's try it over here with the, how do you say, with the crystal violet. The only problem is I need a container. Embarrassing. I don't have one here. So what I shall do is I'm going to, okay, I'm really going to improvise now. You don't mind? Okay. Look, I'm going to, you see, because I didn't, many of the things that I'm doing here, I didn't plan ahead. So for example, also not that I'm going to mix it with alcohol. So I'm going to, this is going to be my little container for the alcohol. Yeah, I've got a bottle for alcohol here, and I've got a bottle of for water. Yeah, so that's just so that you understand this a bit better. Let's lift the crystal violet, and I'm going to dry this a little bit, and let's try to remove the water here, and add a little bit of alcohol. Oh, see how the alcohol reacts. Look, yeah, it turns quite blue. Okay, so this will also de-stain the specimen a little bit. Are you serious? The hour is already over? Thank you very much. I just received again your questions. This went so fast today. And actually, I wanted to also show you a couple of other specimens. So I'm not finished. I want to do this a little bit more. Okay, and what I have to do now is the following. Maybe, I don't know, maybe you've seen that it did shrink a little bit, and the reason is because the alcohol has taken away some of the liquid. So what I'm going to do now is I'm going to add a little bit of water, simply to so let's add a little bit of water so that the cells start to reabsorb the water a little bit, and then they don't shrink quite as much. They will expand a little bit this way. Yeah, let's just give it a try, I would say. We're gonna see how well this works. The cover glass is a little bit wet on the top, so here we go. Let's add a little bit of water here. So, and maybe it brightened up a little bit. I don't know. Okay, let's see. So that is crystal violet now with, where is the microscope? Where's the microscope? Scope. Here it is. Okay, it does look a bit brighter. Yeah, it does look a bit brighter. Yes, now at least we're able to see something. It's still significantly darker than the methylene blue. Ah, look. And what we see on the inside here, okay, yeah, we see the contents of the cell. So the cell wall is on the outside, yeah, and on the inside is what's ever left from the cell, possibly also shrunk together a little bit, the cytoplasm, because as it lost water due to the alcohol treatment, yeah. So because yeah, on the outside here, what you would is you would have the cell membrane, yeah, which is very thin, actually so thin that you're not even able to see it with a light microscope. And on the outside here, you have the strong cellulose cell wall, which gives the plant cell its structure. Okay. The cell wall you're able to see fairly easily. Yeah, and the cell membrane itself you're not able to see, but you're able to see the border from the inside of the cell here. And the reason why it's so shrunk together is because of water loss. Yeah, because of the alcohol, it has basically pulled out quite a bit of the water. Yeah. So I'm just looking again here. So far, no questions. So okay, yeah. So let's have a quick look again if something happened over here. That's again the methylene blue. Yeah, it's 1 hour almost, but I do want, folks, I do want to try something still, and I really wanted to test this, and this is going to be a short one. What I would like to do, and this is a dangerous one now, because I'll be using a sharp razor blade. Let me see, where do I have, if I cut myself and start bleeding, I'll just put the blood under the microscope. Okay. So cross section of some wood. Let's see if this, yeah, the first one here. It is of course difficult. You have to. This is really a question of luck a little bit. You see, you don't need a lot really. Let's give it a try here. Maybe the first couple of layers you just want to scratch away, and then you try to really shave off. It's actually easier to cut it like this, right? With the fibers than across. But actually, if you cut across, sometimes I was able to see more structural detail. But if it really doesn't work, did I cut myself? I almost cut myself. A little bit of blood maybe. No, almost nothing. Yeah. So yeah, this is pretty dangerous stuff what I'm doing here. Okay, let's try it. This is easier to cut, and let's try to shave off a couple of pieces here. Because wood has the advantage that it is basically contains a lot of empty cells, and yeah, very little, but too little. It's already coagulated. So let's move all of this together, and let's give this here a SAFRANIN stain, because SAFRANIN apparently is very good for cell walls. So let's try to give it a little bit of wood. It might take a little bit longer maybe for the wood to actually absorb the stain. I don't know. So a new tip. Okay. So here we have to again remove the stain carefully, because otherwise the background is going to be too dark. Here we go. And then I'll try to answer a couple of your questions. Add a bit of water again. This is not a washing process. I think that's gonna be enough. A cover glass somewhere. And yeah, maybe we're able to. Yeah, again, when I'm doing these live streams, everything is a little bit very fast and quick. A little bit more just a demonstration, and if you want to do this maybe properly, you want to take yourself a little bit more time. Okay, blood might be a bonus. See, it's yeah. Look, we're lucky. I'm gonna put some blood under the microscope. So very quickly before the blood coagulates. So here, that's the too thick. Where is this? Where is this? Here we go. Ah, nice. Okay. Ah, I got some cross sections here. Beautiful. Very nice. Yes, SAFRANIN and wood I declare as being very beautiful looking. Look, that's nice. Okay. So that's basically the cross sections, right? The ones that were so difficult. And these are the longitudinal ones that were easier to cut. So you see how the cells are actually quite long. Actually, this is actually better than I expected. This is, yeah. So, and yeah. So that's the longitudinal cross section. Yeah. And the longitudinal section, not longitudinal cross section, that would be a contradiction. Also the longitudinal one. And here I've got overlapping. These are the ones, yeah, with the cross section. Looks actually pretty nice. Let's go up a little bit with the magnification. Okay, so this is how it looks like. Yeah. Yeah, kind of. Yeah, because it's fairly thick. Yeah. So let's do the following then. Okay, I don't know. Is this possible? Let me quickly try. I don't know if I'm able to do that. Okay, now we have to hurry up a bit. This might be already a little bit too coagulated. No, it works. Yeah, that's more than enough. More than enough. Quickly before it completely dries up. Yes, nice. Okay. Oh, what a crazy live stream. Okay, let's put some blood under the microscope. I've done this already, by the way, quite a few times already in the past. Made some videos. So we need to, here it is. Where are we? Here we go. So, they're already pretty dried up. That's a pity. So, they're kind of dried up already. Let me increase the, here we go. So that's basically the red blood cells now, okay? And let's do it in DIC because they kind of look a little bit better this way. Yeah. And yeah, the thing is, the blood already started to dry up and to coagulate. So for this reason, they don't quite look as nice as they used to. But yeah, here we go. And that's a DIC. That's why it looks a little bit bluish. These are not the natural colors, right? Yeah. So, and one of the nice, I mean, you gotta be patient, and you can do some time-lapse videos. Then you're also going to find some white blood cells moving around, right? And yeah, and this is a very nice sample here. Do you see over here? It looks a little bit like star-shaped. So when the red blood cells, when they start to lose water because they dry up, then they actually start to collapse, and then they do not look like donuts anymore, but actually like they appear to have spikes. Yeah, so that's a natural process that happens when the red blood cells start to dry up. Yeah. Red blood cells, you can have a debate: are they alive or not? They do not have a DNA. They do not have a nucleus. Yeah, so in that sense, they're just empty bag, not empty, no, they're completely full with hemoglobin. So there are bags of hemoglobin, but they do not divide on their own anymore. Yeah, they were formed, of course, by cell division, but in the process they lost their nucleus because the contents of the cell is completely needed for to carry hemoglobin. I don't know, honestly. Blood and crystal violet. Blood and crystal violet? Maybe. Crystal violet is too dark. Actually, I wanted to try SAFRANIN, but I don't know if this is still working. No, see, no. Yeah, so what I'm going to do is I'm going to add... I'm going to try this. Let's try the following. I'm going to show you. I've lifted it now again, but I'm going to add Löffler's solution. Actually, because I wanted to try SAFRANIN, but I know that Löffler's actually does work. So let's put this on here. Okay, and okay, let's see. This is a little bit now. I did not mix it very well, unfortunately. But where is the scope? Yeah, I do want to start answering some of the how do you say, questions as well. But here we already have that, what I wanted to show you, and that's one of the things. Yeah, very nice, very nice, very nice. Basically, I wanted to show this to you because Löffler's solution and other stains. What you see here in the center, that's a white blood cell here, and you see that the nucleus of the white blood cell is lobed and irregular. Yeah, so you refer to it as being polymorph nucleated, and depending on the shape of the nucleus, you can actually just say which type of white blood cell this is, right? And I just did this a very quick. I just dropped a little bit of methylene blue on top of it, right? And you can actually see that there are more of these, white blood cells here. Approximately one white blood cell out of 600 cells. The things that you see in the background, these are the red blood cells, right? They don't stain, but the methylene blue actually stains the DNA. So you see that this one over here has a round nucleus. This one here has a lobed nucleus. So this basically determines which white blood cell you're actually looking at. Yeah. So here as well. Yeah, this was just a quick demonstration. Yeah. See, lots of white blood cells here. Yeah. So yeah, this is a little bit going off topic, but it doesn't matter as long as it's interesting. I think it's fine. Yeah. So what I'm gonna do now is the following. I'm going to quickly go through the questions here. What about blood platelets? Platelets are significantly smaller, right? There is actually a platelet staining protocol as well, but I've not done this yet. Yeah, so I've been staining blood occasionally with methylene blue, like I've done now. Yeah, and by the way, if you're interested in this stuff, I did already make several videos, regular videos about red and white blood cells in this channel here. So this was the practical part. Now I would like to answer some of your questions. Okay, so let me move everything away here, because during the last 15 or 20 minutes, it's also become tradition that I'm going to usually answer some questions here. Okay, some of them are already answered. How the slime molds doing? Yeah, they're doing. Yeah, are now dry, and I've showed them to you. Yep. Okay. Blink twice if the blob is keeping you in hostage. Can you tell us how you make the time lapse of the slime molds? Okay, I can. That's actually quite easily said. The time lapse I've made some weeks ago, some time lapse video. All I did is the following: I took the it's a scary scope view. I've taken the plate with the slime mold. Yeah, I put it on here, right, and I clicked record, and I recorded it for an hour approximately, right. So you get a very large file. And then in the video editing program, I was speeding it up maybe 10 times, 20 times, or 30 times. Yeah, so that's it. Yeah, a little bit of contrast enhancement. So the real thing that really, the thing that really kind of takes a little bit of work and planning is which part are you looking at? Yeah, under the microscope, so to get the composition a little bit right and the lighting and so on. But actually making it was very easy. Yeah. Also put some dried slime molds in the freezer. Yes, I'll do that. I don't know. Yeah, I see no reason why they should not survive this. Yeah. Generally, when cells are moist, when they contain water, and when you freeze them, then this can actually damage the cells because freezing water expands, and this breaks open the cells and the cell dies. Right? The cell freezes, the water expands, the cell pops open because of the expanding frozen water, right? But if they're dry, then generally, I mean, this is actually what is also used for preserving. It's called freeze drying, where essentially while you freeze it, you're applying a vacuum to actually take away the water. Yeah, so this does work. So grateful for your advice to get a Zoom stereo. It's magical. Thank you. Okay, I'm happy. Yeah, I don't remember anymore that I recommended one, but I generally do that. Yeah, I've made a time lapse of frog spawn development. Are you interested? I mean, honestly, if I'll send it to you. Honestly, if you want to share some videos or pictures, please feel free. Send them to me, but you have to tell me explicitly that I'm allowed to show them. Okay, in the live stream. Yeah, because I do sometimes get pictures, but I don't know if it's meant to be shown or not. But if you have anything to contribute and you want to send them to me, it's really no problem. Okay, just tell me if you want me to show it for copyright reasons and so on. Yeah, I would like to study my DNA. You can extract your own DNA pretty easy. I also made videos on that on how you can actually observe your own DNA even under the microscope. No, DNA is actually too thin to be seen, but because there are so many other things, proteins and other things sticking to the DNA, you can actually see, if you're very careful, you can actually see the string-like nature of DNA even under the microscope. Yeah, I asked it before, but I forgot. How much did you thin the PVA with water? This time I will write it down. I think I added around, honestly, I did not measure it out really, but I think I added around 15 to 20% water. And I will tell you why it does not matter so much, because the PVA glue will change its viscosity and thickness anyway as it dries. Yeah, so it's not going to stay constant anyway. It's just going to flow properly, but just the regular fresh PVA glue is way too thick. Yeah, so you have to. Yeah. How much does the SAFRANIN cost? This here, I think I paid only a few euros for that. Seriously. Was it 3 or 4 euros? I mean, now with I also had to pay some import tax and so on. Yeah, but I mean, if you it really was not expensive, really. Okay, yeah, and I mean, I'm not sponsored by them. I just have bought already in the past several things from them. Just go to the webpage of Brunel Microscopes. It's a British company. Yeah, and they pretty much have everything that you want to have. Yeah. I use SAFRANIN with glycerin to stain pollen. Okay, yeah, that's see, maybe next week. Yeah, I predict we won't have any demonstration effect. Probably, luckily, we didn't. Yeah. Has with saffron, I don't know. I mean, the saffronin. Look, there. Now I have to speculate. I know there's this plant that has this saffronin thing in there, which is very expensive because it's also used in food. But I can imagine that maybe this one was synthetically made. Yeah, because in that quantity. Yeah. So I can imagine that these stains are actually synthetically made. Yeah, there were no soot marks. Yes, that's that's why I'm not using a candle, for example, because that produces a lot of soot marks. Yeah, Maybe the suit problem during the heat fixing is also due to the wick being too long. That's an interesting point. That the soot actually comes from the wick and not from the alcohol. Yeah. I just wanted to thank you for the cardboard slide holder idea. Saved my day today. Cardboard slide holder idea. I think cardboard slide holder. Okay. I don't. Was this the test tube holder? Because I demonstrated this once, or this cardboard slide holder. Maybe you can use this cardboard to hold the slides. Yeah, okay. Yeah. Could you not add a diluted drop of safranin on the? I did that. Would you use Canada balsam at this point to make a permanent slide when it's dry? Why not? I've not used Canada balsam before. I'll be honest with you. Because I've got other mounting media that actually also work fine for me. Yeah? Canada balsam has the disadvantage that essentially the specimen must be completely dry, right? Because of the solvent that it contains. Yeah. But I think I see no reason if the specimen is dry, drop a drop of Canada balsam on top of it. But I don't know if it's still very easy to get these days. Yeah. Could you do a second stain? Yes, I did that. Yeah. Substitutions for safranin. You see, to stain red, is this the thing, or generally to stain? Well, the thing is normally the stains react with certain chemicals inside the cell, and it depends really what you want to stain. Yeah. So sometimes I would say there is probably not a substitution if you want to stain specific things. Yeah. Is there some simple way to know what to avoid with immersion objectives? Don't crash the immersion objective into the slide, obviously, right? What my suggestion would be is don't over clean. That's also important. And when you wipe it off using the tissue paper, do not apply pressure, because if there is a little bit of, I don't know, dust or dirt on there, then you might end up scratching the objective. Yeah? Yes, I tried that, how staining affects phase contrast. I guess you can mix stains that target different chemicals. Yeah, that's exactly what I meant as well. Yeah, a little biology, the difference between the cell wall and the cell membrane. First of all, both plant and animal cells, as a matter of fact, all cells have a cell membrane. It's very thin, and it allows only certain substances to go through. Because so it's kind of the boundary of the inside and outside of the cell, and the cell wall provides sufficient strength to the cell, and specifically plant cells. Yeah, plants don't have a skeleton, so they still have to grow up into the sky, and they do that because they have strong cell walls. Yeah? Heat fixing before applying methylene blue, I did not try that now, but that should of course also work. There's no reason why it shouldn't work. Yeah. Why not use the hand microtome? You can use it. I have used it before, and it was simply for speed and practical reasons. Yeah, a staining bonanza, yes, yes, finally caught me live. Can you put some alcohol after the water as well? This will decolorize it. Yes, I have tried that. I was wondering, since I watched him do the cheek cells, he dried them with a small torch on the slide and then put them. No, careful, please. That's actually important. So maybe it's good that you're asking. It is do not heat up the slide when the cells are still wet. Okay, you can warm them. That's fine. Okay, but not drying. What I've done actually by heat fixing it, the slide must already be dry. That's really important. And then I heated it up so that the cells started to stick to the glass. If you heat up something, not just warming it, okay, but if you really heat it up when it's still wet, then what's going to happen is that the cells are going to boil apart. There is steam pressure being formed, and the cells might pop open and boil apart, right? So don't only heat them up when they're really dry, so that there is no steam pressure from inside the cell to destroy the cells, okay? So you can try to carefully warm it, sure, that's not a problem, right? To speed up the drying process, but as soon as you really heat it up, then you're boiling the cells, and then they don't stick anymore, but they actually break open. So that's why it's actually good that you're asking this for me to give me the possibility to clarify this. Thank you. Paul researched it. Safranin at Brunel, 1 gram costs £4 without tax. Okay. But that is essentially 1 gram. Yeah, 4 pounds, maybe it's around €5 or so, right? And I only took 0.3 grams of that, and I made a whole bottle. Yeah. So the bottle's gonna cost you more. The bottles are gonna cost you more than the stain. Yeah. So this is 30 milliliters, 0.3 grams, and yeah. So it's plenty. Yeah. So saffron is derived from saffron. Yes, I'm just checking. I'm now just going up the comments section. Okay. Okay. Going up the comment section. Comment section. So far, no questions. What about the platelets question? I'm going up the comments question. Okay. Cut more. Modern art. Yes. Okay, let me quickly go up. Yeah, I did a little bit of research where I was kind of wondering where the pencil sharpener would be. That's an interesting idea, pencil sharpener. Okay. No, I did a little bit of research. I was kind of wondering, where did the people actually in the past, when they invented the microscopes, when did they actually start discovering the stains and so on? And what I found out is that at that time, a lot of people used stains that were used for the textile industry for staining clothing, right? And apparently they tried out the different stains that they were used for actually making colorful clothing. And this is how it actually got transferred somehow, yeah, to microscopy. Yeah. Oliver, I'm using a Swift 380T trainer. Is there a possibility to increase the intensity of the light? I mean, honestly, if you want to, it's a little bit risky. What you can try to do is you can of course try to a little bit on the do-it-yourself style, maybe exchange the LED, okay, and maybe get a brighter one, or open the condenser a little bit, yeah, yeah, or you have to work with the exposure time, indeed, yeah. But it's always like this. If you go up with the magnification, right, then of course you're also going to lose more light, yeah. Yeah, I think that these were pretty much all of the questions already. Okay. Dropper bottles, thank you. About €5. I mean, look, I can show this to you. Again, I'm not. Let me just a second. Again, I'm not advertising this anything. But I bought myself. I mean, there's so many to choose from. But if you're interested in dropper bottles, I got this over Amazon. I'm not recommending it or not recommending it. It's just the one that I have, and I've bought myself a new set of dropper bottles. Yeah, we even included some of these things here. You can choose all sorts of sizes that you want. Yeah, there's some stickers in here. Yeah, and that's basically how they're delivered. I decided to use the 30 milliliter ones for staining, but you can, of course, whatever you want. Yeah, I think it's kind of nice because this foam in here actually acts a little bit like a holder as well. Yeah, so when these are the fresh bottles, the ones that I did not use yet. And those other bottles, I just keep them in here, yeah, and put them in here like this. Here, I just basically printed out the label, and then I put a broad. You don't see it, but there's a broad tape over the whole thing. Yeah, a thick tape. Yeah? So we can just put this in here again, and yeah, and. If you are from Germany or in the area from Germany, I mean, I don't know, there is this ink that's used in schools quite a lot in Germany, Austria, and Switzerland. And it's this blue pelican ink that's used in fountain pens. This actually also works. So I've been also doing a cheek staining using this one. However, it has one disadvantage, this ink. It loses color if you heat it up. So put a drop of this fountain pen ink into some water and boil it, and the color's gone. Yeah, so that's probably done deliberately unstable. So it's not temperature stable, the ink. Yeah. But that's essentially that's how I keep the stains. Huh? Yeah. So the tomato section. The tomato section. I promised you to do this tomato section. Actually, I prepared a video. What time is it? It goes so fast that I'll just do the tomato section just because I promised you. Okay, I totally forgot about that. That's actually, many of you already know how to make onion, and then because we can also see the cell wall, and I also would like to try to stain it, not knowing if it works. Many of you already know the onion skin being a standard specimen, right? But you can also actually put tomato cells under the microscope. Okay, and it's really self-explanatory. I'm just gonna put this now away. It's really self-explanatory. It's really nothing for me to say here. All you gotta do is just watch what I'm doing. Okay, you take the back side of the knife. And okay, you get the idea. You get the idea. So, by the way, I've already prepared a little, I don't know, 3 or 4 minute tutorial video on how to do that. Now you have to do the following. You have to take the whole thing here. I'm talking now out of experience. And you have to put the whole thing on the slide. And the reason is because if you cut it first, the skin is going to start to roll up. So what I want is I want to make sure it's flat on the slide first before I cut it off, so it won't roll up. And then I nearly forgot about the tomato section here. So it's still a little bit thick here on the side. Okay, if it's too thick, then I won't be able to put a cover glass on top. So now it works again. So that's nice. I need a tissue paper to clean up the whole thing here. Okay, let's do the following. And yeah, I'm going to put it without cover glass or anything, just to show you the cells. Let's put it under the microscope. Let's take this away. We need a much lower magnification. So, and it's too bright. Where is it? Here it is. It's still too. Here we go. Let's turn off the arrow. Yeah, and these are the cells of, basically these are the cell walls rather of the tomato. Yeah. So you see, a specimen's got to be sufficiently thin and sufficiently transparent, and then you're able to see again here the cells and the cell walls. Yeah? So this is actually quite a quick and fast, how do you say, microscopy activity that the teachers can do with students in a classroom, for example. You can try it also at home. But what I would like to try now is I would like to actually do a little bit of staining now, just because we still have maybe a minute or so. How am I going to do this? I have to cut this. I want to cut this because I want to use 2 different types of stains. So let's try like this. Let's see if I'm able to move this over without actually. Yes. Okay. And and now I need the stains again. And I don't know which one should I use. Crystal violet. Should I try crystal violet? Yeah, I'll try crystal violet because it's stained very strongly. So it's the crystal violet. And the SAFRANIN I did not try on the tomato yet. So Löffler SAFRANIN. Okay. So the crystal violet I'm going to remove again with the this really stained a lot. So I'm not seeing a lot because I'm a little bit concentrating now. Up. I've not tried this yet. I just want to tell you, yeah. I'm also going to remove this here a little bit. Okay, a little. So I'm just going to do the following. I'm just going to drop a cover glass on top of each one of them. And just to check how it looks like. Okay, but it seems to be flat. Okay, yeah. So let's have a look. Okay, so this is basically the tomato skin under the microscope, and let's see if it has stained. Here we go. Oh, this is very, very dark. Yeah, same as before. You'd probably have to properly wash it. And what about the crystal violet? This is so dark, I don't see anything. Okay. Yeah, that's how it looks here. Well, I don't know. That's actually quite interesting. Here we can actually compare the stained versus the non-stained parts. Yeah, what we have here is the cell's contents are also so dark now, but it does look a little bit. Let's do the thing away here. But it does seem to stain a little bit in different colors here. Okay, let's try the other one. Here again. I have to find it. Here it is. Let's close the. It's a little too much magnified, I think. Here we go. Yeah, not so. I have to find a place where it's a little bit better visible. Here maybe. Yeah, here this is also where essentially one part is stained, the other one is not. Yeah. Okay, I guess you get the idea. Let me quickly... have you done a gram stain test tutorial? Not yet. I underline the yet part because what I would like to do is that's a little bit something that I'm also planning out to do, actually to grow some safe. Again, important here is safe bacteria in a petri dish. Then if I've got those, then I can do some gram staining and show you, but I need to get them into pure culture first. Yeah. So for this reason, I've not done a gram staining tutorial yet, but I can do that because I do have the equipment now, or the stains, right? Yeah, I've done quite a lot of, not a lot, but a little bit of gram staining already in the past. But yeah. So, are there any more questions? Do you see Marnik's questions about ethanol, methanol, and isopropanol being interchangeable? No, I did not see that question, but here, as it's asked, ethanol, methanol, and isopropanol. First of all, methanol, I'm not even gonna go near because it's so poisonous. Ethanol is the regular alcohol that I also use for storing samples like this, yeah. And isopropanol can be used also in ethanol. However, isopropanol has the advantage, I think it's more expensive as well, but it has the advantage that it contains less water. For example, if you have an alcohol, a bottle of alcohol, then it will basically attract moisture. And until it reaches, I think, about 96 or 97%. Yeah. So, but if you need a less water, if you completely want to dehydrate something, then after the alcohol you go into isopropanol. I have to tell you, for my purposes, ethanol always worked and it was interchangeable. Methanol, I'm not going to use. Yeah. I don't want to inhale the fumes, and it's simply too dangerous. Yeah. So that's the thing. I mean, when I was working in a lab many years ago, I had to use methanol for some chemical reactions, and it was locked away. So we had to I had to talk to someone who basically had the key to the cupboard, and then I could take out a little bit of methanol. Yeah? So. Yeah, so that's okay. Yeah, I just wanted to yes, all nearly forgot about the tomato. And by the way, I will upload also basically pretty much the same tomato, almost the same tomato tutorial also into this channel. Okay, because this one was a fairly long live stream, and I'm making now short videos about short tutorials that can be used simply for for showing in classrooms or at home if you're interested, and so on. Yeah. Is there a temperature to heat fix the slides if you're not using a flame? That's a question, and I would say, I mean, there are these warmers, but they're not warm enough, I think. I mean, you can, there are these coffee warmers that I've been using, but actually the slide has to become hot. So if you don't want to use a flame, you can actually also put it on a hot plate. So for example, in a stove at home, yeah, that should also work. Not stove in the kitchen, right? So this should actually, and I've done this also before, yeah. So without flame. So I'm going to call it a day. Going to call it quits today after a little bit more than 1.5 hours. I think it's gonna be enough. Okay. So we've been again. That's how it looks like. It's actually also a little bit like modern art. Yeah. I'm going to think again of some kind of a topic for next week. Okay. And as you can see, that sometimes topics simply develop as we go along. Okay. Yeah. All the best. Have a nice week, and hopefully see you again next week. Happy microbe hunting, and bye bye.